All right, so picking up what we just left off, moving on with the rest of chapter 2 is going to be focused on our major biomolecules. So these are carbohydrates, lipids, proteins, and nucleic acids. These 4 categories of molecules are what make up a majority of the mass in all living things. And so we'll talk about some of the major characteristics of each of these types of molecules. We'll touch on some of the major functions that they perform in all living things. And then, of course, these are going to show up again and again throughout the course. So getting this introduction out of the way. Oops. All right. So we're going to be addressing carbs, lipids, proteins, and nucleic acids. Carbohydrates are what we commonly refer to as sugars. Lipids are what we commonly refer to as fats or oils. Proteins, of course, we call proteins. You may also hear polypeptide, but those are technically slightly different. And then nucleic acids, which are made up of nucleotide monomers. As we have a bunch of different examples of molecules that fall into these categories. We'll talk about the monomers, which are the single building block units of each of them, and then some of their polymers, the larger forms that are made up of those monomers. So for carbohydrates, the smallest individual unit, a single sugar, a single carb, is a monosaccharide. Mono meaning one, saccharide coming from the Latin root saccare for sugar. So monosaccharide is a single sugar. Disaccharide, prefix meaning two, dis for two sugars, poly meaning many, so this would be 3 or more sugars. Lipids are made up of fatty acids and glycerol heads. And so those can come together to form different types of glycerides. We'll talk a lot about phospholipids in this class, glycolipids, steroids, eicosanoids. Proteins are all built up of amino acids. So amino acids are the monomers there. And there are 20 different types of amino acids, which can come together in different orders and sequences to form our various types of proteins. And there's lots of different types of proteins. And then nucleic acids are what make up all the genetic material of living things. So our DNA and RNA. And these are made up of nucleotide monomers. So all of our biological molecules have carbon as their main backbone. And like I said, carbon tends to be pretty generous with its electrons. It's not very greedy. So they will only form polar covalent bonds if they are bonding with a more electronegative element, like nitrogen or oxygen. But that carbon and the hydrogens bonded to them don't really provide much of a function. They are really just there for structure. It is these other additional groups that end up forming bonds with carbon, with that carbon backbone, that give these molecules their function, their ability to interact with certain things. And so groups that give similar properties to a molecule are organized into what we call functional groups. So we have some of our major functional groups listed here: amino groups or amine groups, which are common in amino acids; carboxyl groups, which will also show up in amino acids; hydroxyl groups, which is just an oxygen and hydrogen; and then phosphate groups, which can be utilized for energy or for stronger bonds and things like nucleic acids. So these are some of our major functional groups that we'll see showing up, which give these carbon backbones their function and are very specific to different biomolecule categories, types of biomolecules anyways. All right, so carbohydrates tend to be the simplest. Carbohydrates are only composed of carbon, hydrogen, and oxygen, and they will always exist in this ratio of 1 to 2 to 1 in a carbohydrate and a sugar. So if there are 6 carbons, that molecule will always have 12 hydrogens and then 6 oxygens with it if it is a carbohydrate. So that 1 to 2 to 1 ratio always stands true. And so most monosaccharides are 5 to 7 carbohydrates each. That relatively stays true throughout all of them, whether you're looking at glucose, fructose, galactose. Those are all going to have 5 to 7 carbohydrates in their structure, and thus will have the relative hydrogens and oxygens to go with it. And they can either form these linear structures, which are the most common chemical structure we'll see, or molecular structure that we'll see for all biomolecules, or they can form what we call a ring structure. So anytime you see this ring, where we have carbons, 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 sometimes an oxygen punched in there, all making this geometric shape, that is a carbohydrate. And so you're going to see this ring structure show up in some of our other biomolecules. We're looking at like our proteins, looking at amino acid structure and looking at nucleotide structure. You will see a carbohydrate as part of that monomer structure, this ring form. But again, only carbon, hydrogen, oxygen. They're always in that 1:2 ratio. And then the monomers of carbohydrates, the smallest, the single unit, the building block for sugars are monosaccharides, one sugar, monosaccharides. And so the most common polymer, really the most common form of carbohydrate that we're going to be talking about is glucose bonded together into a glycogen. So glycogen is this really large structure of multiple glucose molecules bonded to each other. And this is how our bodies as humans, as animals, store carbohydrates for later. So glycogen is the polymer of glucose that you would find in a human body, and that acts as stored energy. Carbohydrates are accessed as fast, quick, efficient energy for when we need that energy quickly to do work. And so we have glycogen stored in our metabolic headquarters, our liver, that will break down glucose when we need it, or will store excess glucose as glycogen when we have extra. But we also have glycogen in our skeletal muscles or in our muscle tissues in general, because muscles are very energy dependent and very energy hungry. And they need a lot of it to do the work that they do, to move our skeleton and interact with the world and generate force. So we also have glycogen showing up in our muscles. Again, mainly skeletal muscles, if we're looking outside of the liver. And so in plants, they have their own version of stored glucose called starch. Right? So like a potato is literally like a glucose storage organ. It's just full of starch for that potato plant. But in humans, we're only focused on glycogen. For digestion, you'll talk about the others, but as far as how we store our energy, it's as glycogen. Okay. Lipids are our next most basic version of a biomolecule. Again, lipids are what we commonly call fats or oils. Lipids are also solely composed of carbon, oxygen, and hydrogen, but with a slightly different ratio or setup. A lot less oxygen in a lipid. So the oxygen that shows up in carbohydrates is important to mention. Okay? Oxygen is electronegative. So portions of our carbohydrates are going to be polar and thus can form interaction with water and create those hydration spheres that we talked about earlier. Lipids, which have much less oxygen, tend to be mainly non-polar, and thus they are hydrophobic, especially our fatty acid tails. Fatty acids are these long chains of carbon bonded to carbon bonded to carbon, with hydrogens taking up the rest of the available bonds. We only get oxygen showing up at this carboxylic acid group at the end, which is going to form other bonds with other carbons, making the glycerol head. So the fatty acid tail itself, you could basically just consider it is completely non-polar, it is completely hydrophobic. And we have a couple of major types of fatty acids. We can refer to them as either saturated or unsaturated, depending on how many hydrogens are saturating the carbons in that fatty acid tail. And so fully saturated fatty acids tend to be solid at room temperature. And so if you think of any fatty foods that maintain their solid structure at room temperature, like butter and lard, those fats are saturated fats. Unsaturated fats tend to be missing hydrogens on one or more of the carbons in their chain. And so that remaining bond is going to be used to make a double bond with a neighboring carbon. And what that does is it bends the structure of that fatty acid tail, creating space. And I think I have an image. Yeah, so we'll come back to that later. In an unsaturated fatty acid, when we have these hydrogens missing and thus double bonds are created between carbons, that creates a kink, a bend in the fatty acid chain, which makes space between neighboring fatty acids, which means that those fatty acids are going to be more fluid, more mobile. They can move easier. And so most unsaturated fatty acids end up being liquid at room temperature. So these are all of our oils, canola oil, avocado oil, etc. That comes from this double bond that creates a kink. So unsaturated fatty acids can also be looked at depending on where the one hydrogen that is left over is. It could either be cis or trans, and that has a larger role in our nutrition and digestion. So I don't think we talk much about it in this class. So mainly just knowing the difference between saturated, meaning there are no double bonds, it is a linear structure, solid at room temperature, versus unsaturated. There are double bonds, at least one double bond, creates a kink, making space, and thus these are liquid at room temperature, a lot more fluid and mobile. Something else to touch on here. Fatty acids, whether they are saturated or unsaturated, are constantly being manipulated in our cells too. So if we need to have more fluidity in something like our plasma membrane, we can add energy to these fatty acid tails, create more double bonds, making them unsaturated, and thus allow for more movement. So now a protein can have an easier time moving through that plasma membrane and get to where it needs to be. And then we can remove that energy, add some hydrogens, and put it back as a saturated fatty acid. And now it is more structurally stable and solid. So we are constantly manipulating between those. And so most natural forms of fat, or most of the molecules that are going to be formed with fats, are going to involve a glycerol head, which is technically a small carbohydrate, a glycerol head with multiple fatty acid tails attached to it. So if we have 3 fatty acid tails, like what's shown here, this is a triglyceride. If you had 2, that would be a diglyceride. Right? If you had 1, that's a monoglyceride, right? Referring to a glycerol head with fatty acid tails. We do have some other examples of fats that don't follow that same general format of a glycerol head with fatty acid tails, and these are our steroids. So these tend to be used as long-range chemical messengers. Right? So hormones, you do have lipid-based hormones. So these are things like testosterone and corticosteroids, estrogen. Right? These are fat-based hormones that are long-range chemical messengers in your body. But the most common steroid in our body is cholesterol. We have cholesterol embedded in our plasma membranes to make them more stable as well. So cholesterol, although commonly talked about in general news and things as a dangerous thing in your diet and you need to avoid it, yada, 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 that's based on past science that has been debunked. Cholesterol is fine for you. It's just about getting your right balance of nutrients, because cholesterol is an integral part of your biology. You need that in your plasma membranes to maintain their stability. And then arguably the most important type of lipid, at least when talking about how biological things work, is the phospholipid. Right? This is what makes up a majority of all membranes in all living things. Every single thing that is considered alive is going to have membranes made up of these phospholipids. And so a phospholipid is unique from what you would call a diglyceride. It has a glycerol head, it has 2 fatty acid tails, but it also has a phosphate group attached to that glycerol head, with this non-lipid group pointing out as well. And these are polar. And so in a phospholipid, that gives this glycerol head some polarity and the ability to interact with water. So phospholipids in our membranes will have their heads, their hydrophilic heads, pointing outwards to the external environment and inwards to the inside of the cell, because those are both going to be aqueous environments. And then we have our nonpolar, our hydrophobic fatty acid tails pointing towards the inner parts of that membrane, creating a selectively permeable barrier. So in that membrane, we have various types of things being attached to different phospholipids, allowing for things like carbohydrates to stick out or proteins to stick out of that membrane. And so if we have a carbohydrate attached to a lipid, that is a glycolipid. And these are often used for communication and recognition with neighboring cells. Right, you can see those sticking out here. Glycolipids of multiple neighboring cells will form bonds and create interactions, maybe create channels, or used to help create channels, or used to communicate. I guess these are showing my cells, but same idea. Okay. I basically talked about all of this stuff with cell membrane anatomy. Again, we're going to talk more about this in the next chapter, in chapter 3. But of course, membranes are the barriers between cells and the environment, or the barrier between an organelle and the rest of the cell. So recognizing how their structure dictates their function and what those functions are is going to be really important for understanding how things move in and out and communicate, et cetera, throughout our systems. Yeah, touch back in on all of this stuff later. Right. So our next major biomolecule is proteins. So we talked about carbohydrates and lipids. With both of those biomolecules, you will only see carbon, hydrogen, and oxygen show up in their chemical structure, just with slightly different ratios, giving them slightly different functions. Proteins are where we're first seeing a new element. We will see nitrogen show up, because proteins have the functional amine or amino group in their structure. So proteins are all made up of different sequences and orders or numbers of amino acids. So amino acids are our monomers, our building blocks here. And there are 20 different types of amino acids that are utilized by all living things. And each amino acid has the same basic structure. We have a central carbon with one of its bonds being given to hydrogen, one of its bonds being given to this amine or amino group, which is NH2. Its third bond is being given to this carboxyl group, which is COOH or CO2H, however you want to refer to that. And then its last bond is going to differ between each amino acid. So all of our 20 amino acids are different from this very last bond, our R group or variable group. And the bonds between individual amino acids have their own name called a peptide bond. They're formed in the same way. They can be created through dehydration or broken apart through hydrolysis. But we're bonding carbon with nitrogen in this case, and that is called a peptide bond. So we have a peptide bond. Multiple amino acids being bonded together through peptide bonds would create a polypeptide, multiple peptides, right, because of those peptide bonds. And so peptide bonds are unique to amino acids. If you're asked a question about what kinds of bonds hold amino acids together, it's a peptide bond. If you're asked about where would you see a peptide bond, it's in proteins and polypeptides, bonding amino acids together. And so when we're creating proteins, which are the major tool of the cell, I guess I haven't been giving enough dedication to like the function. Right? I mentioned that carbohydrates, their major role is as quick, accessible energy. Lipids, their major role is as energy as well. They are stored energy that they provide more energy per gram. So while our body is at rest, or we only need energy at a regular pace, we'll actually use fats as our energy source instead. We use lipids. But carbs and lipids do other things too. Carbohydrates play a role in communication. Lipids play a role in our membranes. They play a role as insulation, right, as protection. Proteins do everything else. Proteins are what your entire set of genes are used to make. Your DNA is a set of instructions for building these, for adding amino acids together in a particular sequence. And then those amino acids working together are going to perform whatever job that gene coded for. So the reason I have brown eyes and brown hair is because of my genes that coded for melanin production. Melanin is a protein in my eyes and in my hair. Right? So moving on from that, right? So all of our proteins are going to be formed based on those instructions in DNA. Those instructions get turned into this sequence of amino acids. So it tells our ribosomes what amino acids to add together and in what order. And so what comes out of the ribosome is this linear structure of amino acids with peptide bonds holding them together. And we refer to this as our primary structure. This is a non-functional polypeptide, multiple peptide bonds, right? Polypeptide. And a polypeptide is typically the term used to refer to an immature protein, so a protein that is not able to do its function. So in its primary structure, this is a polypeptide. It cannot do its job. It is simply a string of amino acids that do not have their form or function determined yet. And so that string of amino acids is going to start forming hydrogen bonds with its polar portions. So these interactions are going to start naturally creating those magnetic attractions, and that is going to cause that polypeptide to create either what we call an alpha helix, this kind of spiral looking structure, or a beta pleated sheet where they start getting some bends. And this is still an immature polypeptide, right? But we're starting to get some form to our protein. And this is our secondary structure. So primary was the linear chain. Those natural hydrogen bonds are going to create a little bit of structure, either this helix or this pleated sheet. That is our secondary structure, but that protein can still not do its job. Almost all proteins, 99.9% of proteins, are only functional once they have reached their tertiary or quaternary structure. So the tertiary structure is referring to a single polypeptide that has been pushed and bent into this final form. So it typically requires other proteins to help move that secondary structure into its final form, which now has the shape and chemical composition it needs to do its job. Like in this case, we're looking at a heme protein, which is going to be a part of hemoglobin. And that heme protein needs this shape and needs an iron molecule, an iron atom, sorry, an iron ion in this portion of it to be able to bond with oxygen and carbon dioxide and carry those things throughout our blood. And it was not capable of that job until it was bent into this tertiary structure. And we have very strong disulfide bonds holding those amino acids together in this shape. And so again, 99.9% of proteins can do some job or function in this tertiary structure. A lot of them will then be taken further into a quaternary structure, which would be multiple polypeptide chains bonded together. So for hemoglobin, we have 4 polypeptides in their tertiary structure that have all now been bonded together to form one large protein called hemoglobin, which can carry 4 molecules of O2 or 4 molecules of carbon dioxide and carry that throughout your blood via your red blood cells, and thus bring oxygen to your cells for respiration and bring carbon dioxide to your lungs to breathe back out into the environment. But all of our protein fibers are quaternary structures as well, like keratin and collagen, which are going to show up a lot in this class. These are long chains of multiple large string-like proteins working together in their quaternary structure. So altogether, right, you can see those various structures and how they build on each other. Again, for a mature protein, for it to be able to perform its function, it has to reach its tertiary or quaternary structure. In its primary or secondary, it is still considered immature, so it is a polypeptide and cannot yet do its job. Okay. So one of the major roles of proteins that we're going to be looking at is their role as enzymes, lowering the activation energy needed for chemical reactions. So making it easier to build large molecules or break apart large molecules depending on the need. But we have lots of different roles to discuss. Right. So lastly, nucleic acids. Okay. Nucleic acids serve one purpose. They are genetic material, okay, either as DNA or RNA. Right? DNA being the stored genetic material that's passed on from cell to cell or from parent to daughter cell. RNA is used as the intermediate to copy or transcribe the information from DNA and then use that to form a protein. And then some viruses, which aren't considered living, but do share a lot of the functions of living things, can have RNA genomes. So it seems likely that RNA was evolved first or generated first naturally in the environment and then developed into DNA. The difference is a single oxygen in their structure, right? But they're basically the same thing. So nucleic acids are all made up of strings of nucleotides. So proteins are strings of amino acids. Nucleic acids are strings of nucleotides. And we have 5 major nucleotides to recognize. In DNA, we have adenine, guanine, cytosine, and thymine. In RNA, the only difference is that they have uracil and they do not have thymine. So that one difference is going to show up when looking at transcription and translation. So taking DNA code, transcribing that into an RNA transcript. Right? Anywhere where a thymine, you would have expected a thymine to show up for DNA, instead a uracil will show up in RNA. That's basically it. And so we can categorize these as purines and pyrimidines based on how many of these ring structures show up on that nucleotide. And that dictates which other nucleotides they can form bonds with in that double helix structure of DNA. But for the purposes of this course, really knowing what a purine or pyrimidine is isn't very important. What you will need to know is that A forms bonds with T and G forms bonds with C and vice versa. And so then when looking at transcription, if we are transcribing and reading the template DNA and there's an A, RNA is going to have U instead of T. So adenine bonds with thymine or uracil, guanine binds with cytosine, and vice versa. Right. So again, we'll touch more on that in chapter 3 when looking at transcription and translation, but that's the general difference between these nucleotides. And so all nucleotides have the same basic structure. We have a ribose sugar, thus the name deoxyribose or ribonucleic for DNA and RNA. We have a ribose sugar making the center, a hydroxyl group, which is going to bond with the phosphate group of its neighboring nucleotide, creating these strings of nucleic acids. And then a nitrogenous base, which is where we get these purines and pyrimidines. So the nitrogenous base is what is going to be different between each nucleotide, but all nucleotides will have a central ribose, a hydroxyl, and then a phosphate group. And so the backbone of our DNA is from those phosphate groups forming bonds with the hydroxyl group of the next nucleotide. Okay. So that forms this backbone with our nitrogenous bases sticking into the center. And so in DNA, we're going to get 2 strings of these nucleotide backbones that are going to interact with each other and form hydrogen bonds, holding both backbones together and creating that double helix model that we all have come to recognize DNA as. Okay? So we have our ribose sugars with their phosphate groups and hydroxyl groups bonding to each other, creating these golden strings that we have representing our backbones. And then we have our nitrogenous bases creating hydrogen bonds in the center, holding those strings together. And so when we are going to read this DNA to either replicate it and create a new cell, or to read it as a template to create a protein, those 2 strings get unraveled or pulled apart, and then we create a copy based on the complementary nucleic acids or nucleotides. Like I said, we'll expand on that in the next chapter. Just recognizing structure here is important. Okay. And I believe our last molecule that we're going to be talking about in this chapter is ATP, adenine or adenosine triphosphate, adenosine triphosphate. For a long time, I've said adenosine, but adenosine is the more common form and the right form, adenosine triphosphate. So adenosine triphosphate is made up of an adenine and ribose sugar as the base, with 3 phosphate groups bonded to each other. And this is a high energy carrying molecule that can be accessed by any part of the cell, or at least a protein can break those bonds anywhere in the cell and thus release some free energy to power work. And so ATP is the cellular currency, the main energy carrier for the entire cell. Right? All of the energy that is contained in like a carbohydrate or a fatty acid is going to require much more specific enzymes to access that energy. So our cells tend to harvest energy from these larger biomolecules and then turn that into ATP, and that ATP can then be used to power any work anywhere in the cell. Right? It's all based on these phosphate groups. Phosphate is a negatively charged molecule, and it's a polar molecule and does not like being close to each other. And so they are constantly pushing, trying to break apart when they have been forced into these bonds. And so if we break a bond between the second and third phosphate, a lot of energy gets released, and thus work can be done. And so that is going to leave behind a adenosine with 2 phosphates, which we refer to as ADP, and that is constantly being recycled back into ATP and so on. So ATP is being brought somewhere that needs energy. We break off this 3rd phosphate, and now we have an ADP and a free phosphate left over. That phosphate and ADP are being brought back to like the mitochondria, where they will be turned back into ATP using energy from carbohydrates or fats. And so ATP is the major power source for all things that are happening in the cell based on those high energy phosphodiester bonds. So we had our peptide bonds for amino acids. These bonds holding phosphates together are phosphodiester. We have our nitrogenous base that we saw with nucleic acids and adenine. We have a sugar, a ribose, and then a bunch of phosphates making up that structure. And that should be all of the major concepts from chapter 2. This does not guarantee that I covered everything in the chapter, so please do still read through or at least look over chapter 2 and use those learning outcomes, those learning objectives as a guide to your note-taking. As always, if you have questions, comments, or concerns, please do not hesitate to reach out to me or come talk to me during office hours or talk to me during our live lab meetings. And I will see you guys in the next one.